Okay, so moving on to number 2, this one says, during meiosis, double-strand breaks occur in chromatids. The breaks are either repaired by the exchange of genetic material between homologous non-sister chromatids, which is the process known as crossing over, seen in figure 1a. Or they are simply repaired without any crossing over, which is figure 1b. Plant breeders developing new varieties of corn are interested in determining whether, in corn, a correlation exists between the number of meiotic double-strand chromatid breaks and the number of crossovers. Okay, so we've got one figure here, figure A. You've got a double strand break in chromatids that are repaired by crossing over. So we do go over the process of crossing over. As you can see here, they've swapped genetic material. And then B is if they just get repaired without any crossing over. So you've got the breaks, but there was no exchange of genetic material. So you've got two options here. Okay. Now, it says, using specialized staining and microscopy techniques, scientists counted the number of double-strand chromatid breaks and the number of crossovers in the same number of meiotic gamete-forming cells of 6 inbred strains of corn. So we've got a table here. So table 1 says the number of chromatid double-stranded breaks and average number of crossovers in inbred strains of corn. So we've got 6 strains of corn, and then you've got number of double-stranded breaks, and then average number of crossovers. Okay. Okay, so we can just dive into A, which says the double-stranded breaks occur along the DNA backbone. Describe the process by which the breaks occur. So they're telling you that these breaks are happening from the backbone, and obviously because they need to be able to exchange that material. And they're kind of just asking you what is happening when the strand breaks, okay? So if we think about the backbone and we think about the actual structure of DNA, it's a sugar phosphate backbone, and then you have the bases on the inside. So I can roughly try and draw, go like this. That. And then you would have your bases here, that, right? So then this is your backbone here, and then these are your bases in the middle. They're saying that the break is happening in the backbone, okay? So we are cutting this part. We're not cutting down because that would be breaking the hydrogen bonds and separating the two strands. We're breaking the backbone. So what is this doing? Well, the backbone is made of sugars and phosphates. So they would have accepted 2 answers for this one. You could say that you are breaking the covalent bonds between the sugars and phosphates, or you can say that a hydrolysis reaction occurs between the sugars and the phosphates, okay? So you could have also specified that this is what's known as a phosphodiester bond, but just saying it's a covalent bond was enough. So this one's actually pretty straightforward. So then for A, I would just say that the covalent bonds, or specifically phosphodiester, you didn't have to say that, but covalent bonds between sugar and phosphate break. Okay, so that's really all you had to say for these. And you could have also mentioned, you could have also said a hydrolysis reaction. So covalent bonds breaking, it happens through a hydrolysis reaction, so you could have said that as well. Covalent bond between sugar and phosphate break / you could say hydrolysis. But you would have to say that it's an hydrolysis reaction that separates the sugars and the phosphates. Okay, so that was pretty straightforward, I feel like. Okay, B is our big graphing question. It says, using the template in the space provided for your response, construct an appropriately labeled graph that represents the data in Table 1 and allows examination of a possible correlation between double-strand breaks and crossovers. Based on the data, determine whether corn strains 1, 2, and 3 differ in their average number of chromosomes. Okay. So they would have given you like a whole sheet of graphing paper for this. When they say they're giving you a template, they're not telling you what the graph is supposed to be. So they're not telling you if you should do a bar graph or a scatter plot or a line graph or anything like that. That's for you to decide based on what the variables are. All they're giving you pretty much is graphing paper. So I don't have the template on me right now, but it doesn't really provide you with much. So I'm just going to roughly draw this graph. So it's really important to go back to our variables and to think about what would be the best kind of graph for this. Now, the key thing here, if we look, we've got strains of corn, and then we've got 2 measurements for each. And there are 2 measurements of 2 different things, okay? We have number of double-stranded breaks, and we have average number of crossovers, okay? So for this one, it would not really make sense to do a bar graph, right? Because then what do you put on the y-axis? If I do a bar graph, the x-axis has to be strain of corn, and then the y-axis, you can't choose one or the other, you have to graph both. You also can't do a double bar graph in this case because, again, what's your y-axis going to be? Okay, so the key for this one, and a big giveaway for what type of graph you want to do is you're looking for a correlation. You're looking to see if one variable affects the other. You want to see if the number of double-stranded breaks correlates or affects the number of crossovers. Typically, when you do that, you're going to want to do some sort of scatter line plot, okay? So in this case, we're going to do a scatter plot, and we're going to have one variable on the x-axis and one variable on the y-axis. For the strain of corn, to identify it, we're just going to put a label beside it, okay? You should not do, there's no way to do this with a bar graph. Definitely not a box and whisker plot for this one either. A line graph is going to be the best for this one. It really just depends on what variables you have. You have to think kind of logically about these. Okay, so if I were to do this, let me start to draw my graph. I'm going to put the number of double-stranded breaks on the bottom. It technically doesn't matter which one you do. They probably would have marked it okay for either of them, as long as you have the 2 variables on the separate axis. So I'm going to draw a big graph like this. Make sure you can see all of it. There we go. Alrighty, so now I'm going to start to put my labels in. When you do these graphing questions, it's really important that you label everything because that's part of the marks. So I told you that I was going to put number of double-stranded breaks on the bottom, so I'm going to label that here. So you can put number double-stranded breaks on your x-axis, and then your y-axis is going to be your other variable, which is average number of crossovers. Okay. So we'll put, please write these in full. I'm just trying to save time. Number of crossovers. And it includes the metric for standard error, so you also want to include that as well. For these ones, there's no unit because it's more so a count. If there were units, you have to put the units into the labels, okay? So if it was measuring it in milliliters, you have to put milliliters beside. That's not for you to determine. It will tell you in the table what the measurement is. Just copy exactly what it says. When in doubt, just recopy the exact same label. Okay, so it's plus or minus 2 standard errors. Okay, so I'm just going to put that as well. So plus minus 2 standard errors. Okay, so for these ones, they don't really need you to do like a top graph, or sorry, like a top title for your graph. Just label your axis. And then the next important thing that you're going to do that gives you marks is determine your scale. So your scale is going to be really important. Let me just zoom this out a little bit. Okay. Just want to be able to see the whole thing. Okay, so the scale is going to be really important, and I give the same tip every single time when we do these. Obviously, make a mindful decision based on how much space for your graph paper you have. But what I typically do, in this case, you need a range on your x-axis and your y-axis because they're both numbers, right? So for my number of double-stranded breaks, I always look at the lowest number and then I look at the highest number, and that's going to give you the relative range that you want to use. The lowest number is 325, the highest number is 710. Okay, so you're going to be having points between 325 and 710. I would probably then proceed to go up by 50, for example. Again, try and be mindful of the space. So they usually, I want to say, give you like a 30 by 30. You don't have to use the whole thing, but also make sure that it's not tiny in the corner. Just try and use a good amount of space on the graph. So in this case, we're going up to probably around 750. So what I'm going to do is go 0. Let's put 100 here. We'll do 200. 300, and then we'll put 800 here. Okay, I know I wrote that they're going up by 100, but what you're going to want to do then is make ticks in the middle. So yes, 0 to 100, and then this is going to be 50, that'll be 25, and that'll be 75, right? You don't have to write all the numbers, but the scale is going to be important. So I'm going to put 3 ticks in between these just to give me a little bit better reference. Again, just make a logical decision based on how much graph paper you have. Okay, so that should be good. Now we do need an axis as well, or a scale for our y-axis. So again, do the same thing, look at the lowest number, then look at the highest number. My lowest is 11, my highest is 19.5. So I'm probably going to go up by 1. That would make the most sense. If you obviously don't have 20 squares, don't go up by 1, but you'd likely have more than 20 squares. So what I'm going to do then in this case, we're going to go from 0 to 20. So this one's going to be 0 right here. I'm going to go up by, actually I'll go up by 5s and then I'll draw ticks. 5, 10, 15, 20, and then I'll draw the ticks in between. Okay, so you do get marks for what I just set up. So your axis titles in a proper scale is going to be really, really crucial. Now we're going to have to actually plot them. So that's, only like a third of the actual mark. So we're going to go through and we're going to plot these. And I'm going to show you how to do standard error as well, because that's going to be really important. Okay, so let's just start off with number 1, which is 710 and 19.5. 710, 19.5. I'm going to do this. Let's do this in green. So 710 would be around here, and then 19.5, we're going to drag it all the way up to around here. Again, this would be much easier if you had graph paper to follow the lines. I'm doing this roughly. Okay. And then one thing that we have to remember to include, so that's supposed to be 710 and 19.5, pretty close. Is they include standard error here. So because they include standard error, you have to put standard error in your graph. You can't just ignore that. There's only standard error for one of them, which is why you want to put this one on your y-axis. It's just going to make it a lot easier. The standard error in this case is 0.5, so you're going to want to draw your error bars 0.5 up and then 0.5 down. The errors are different for each of them, so just be mindful of that. So you're going to go up by 0.5, so this is that 19.5. You should draw your standard error bar up to 20 and then down to 19. Okay, again, they're not going to measure this with a ruler. Just make sure it's like, somewhat accurate. As long as it looks okay, then it should be fine. And then the last thing that we want to do for this one is, because we're going to have 6 points plotted, you need to label these, right? So all I'm going to do for these ones is probably just put the number beside them. So this one is number 1. I'm going to do it in a different color. This is number 1. Okay, that's all you have to do. Now, the next one, number 2, is 650 and 18. Okay, so 650 is around here, 650 and 18. Okay, we've got 650 around here, gonna drag that up to around 18, which should be like here. Okay, and then your standard error is 0.7, so you're going to go up by 0.7 and down by 0.7. Should be a little bit longer than the error bars you drew for number 1, but again, just try and make them as close as possible. And then you're going to want to put your number beside it so you know which one is which. Okay, number 3, 617.5. So 600 is right here, 17.5 would be around just right below this one because that one's at 18. Your error bars are 1, so those are pretty large error bars. These should be the largest ones yet. So this was supposed to be at 17.5, so you should have the error bar go up to 18.5 and then down to 16.5, just as a frame of reference. So go up to 18.5, should be around here, and then actually I should make this one longer because it should be longer than the ones from number 2. Okay, and then this is number 3. Alrighty. Number 4, 5, 10, and 16. 5, 10, and 16 should be around here. And I believe there was a 1 error bar. Yep. So up to 17 and down to 15. 15 up to 17. And then this is number 4. Almost done. Number 5. 425 and 14. 425 is right here, and 14 is right here. And there is a 0.5 error bar for this one, so a pretty small error bar. This, oops. It's number 5. And then our final one, number 6, should be 325 and 11. 325 is right here, and the 11 is here. And then it's a 1.5 error bar, so it should be like the longest error bars we have. And then that's number 6, I believe. Okay, so that is our graph. So this is everything that you should include. Make sure you put the labels or you're not going to know which one is which. Make sure you include your error bars, all your units, and everything like that. So you should get marks for your scaling, you should get marks for your labeling, and then you get marks for accurate plotting. They're not, again, looking for it to be, super, super specific by the millimeter. As long as, it's accurately labeled, then you should be fine. Okay, so now that we've drawn this, let's reread the question because I think there was a second part to this. It says, using the template in the space provided, construct an appropriately labeled graph. Based on the data, determine whether corn strains 1, 2, and 3 differ in the average number of crossovers. Okay, so they are looking for whether corn strains 1, 2, and 3 differ in average numbers of crossovers. Okay, so let's do 1. Average number of crossovers, so 1, 2, 3. Now, for this one, these questions are pretty specific, and this is why you have to practice these, because they're asking you something fairly specific. For this one, they're asking if there is a difference between them. Sometimes they will say like significant difference between them, but either way, obviously the numbers are different. That's not what they're asking. So you have 19.5, 18, and 17.5. Obviously the numbers are different. That's not what they're asking you here. You have to consider the error bars for these ones, because when it says 19.5 plus or minus 0.5, what it's really saying is it's between 19 and 20. Okay, 18 plus or minus 0.7 is going to give you 17.3 to 18.7, okay? And then 17.5 plus or minus 1 is going to give you 16.5 to 18.5. Okay, so they are not specific numbers because they have the standard error here. They're a range, okay? And for two things to be considered different from each other in terms of value, their ranges cannot overlap. Okay, if they overlap, then they're not really different from each other. So we have to make sure that all three of these don't overlap with each other to make sure that they're different. So it looks like 1 I would consider as different from all of them because none of these are in the 19 to 20 range. However, it's looking like, yeah, 2 and 3 are going to overlap, right? 17.3 to 18.7 and then 16.5 to 18.5. Those are overlapping with each other. So their ranges overlap. Therefore, we cannot say that these ones are different. Okay, they very well could have the exact same value when you do a repeat measurement. Their ranges are overlapping. So that's when you have to say that it's, there's no statistical difference. So for this one, determine whether corn strains 1, 2, and 3 differ in the average number of crossovers. So for this one, I would say, this is B. We said that 2 and 3 are not statistically different. Let me see. I think this was only worth 1 mark, so you didn't really need to justify it. And then you had to say, however, strain 1 was significantly different. Strain 1 was higher than both. Okay, so whenever it asks you difference, you need to consider the standard error. You always consider the standard error when it's given to you because the standard error is referring to the fact that you have a range of values. It's not one complete value. It is a range of values. You don't have standard error for the number of double-stranded breaks because you're counting those. Average number of crossovers, you're going to have a range because the average may vary. So just be careful and make sure you look, and that's going to determine whether they're statistically different or not. Okay, so that's B. C says, based on the data, describe the relationship between the average number of double-stranded breaks and the average number of crossovers in a strain of corn analyzed in the experiment. So they're just asking you to describe the relationship. And for this one, look at your graph because you already pretty much graphed the correlation. Now, for this one, there's a very clear pattern for this. Hopefully you can see if I were to draw my line of best fit or like the average line, I'm going to do this very roughly, it would go like this, right? This way. So as the number of double-stranded breaks increase, the number of crossovers also increase, right? The one that has the highest number of double-stranded breaks, number 1, also has the highest number of average crossovers. The one with the lowest number of double-stranded breaks has the lowest number of crossovers. And so you're going to get this line here, and this is what we would call a positive correlation or a direct relationship. A positive correlation is always when the increase of one increases the other. A negative correlation, they're either going to ask you if it's positive or negative. There's like nothing else. Negative relationship is when one increases, the other one is going to decrease. If this were a negative correlation, it would look like this. Okay, but it's not. It's a positive correlation going this way. So that's all you had to say for this. Based on your graph, you would just say that this is a positive relationship or what we'd say like a direct correlation. So C, say positive relationship. That's all you had to say for this one. Typically, when they ask you these, they're either going to be positive or negative. And again, that might not be something your teacher explicitly goes over, but it is something that you're going to have to practice for these ones. Okay, the last one is D. Okay, so D is going to be a little bit longer. it says crossing over, which you can see in figure 1, creates physical connections that are required for proper separation of homologous chromosomes during meiosis. A diploid cell with 4 pairs of homologous chromosomes undergoes meiosis to produce 4 haploid cells. Crossing over occurs between only 3 of the pairs. Predict the number of chromosomes most likely to be present in each of the haploid cells. Provide reasoning to justify your prediction. Explain how plant breeders can use this information in Table 1 to help develop new varieties of corn. So let me just move this down so that we can see the question and the answer at the same time. Okay, so this is obviously going to be like our application-based one. That's going to be a little bit more difficult. So you have a diploid cell with 4 pairs of homologous chromosomes. So I'm just going to draw that for visual. So let's go like this. That's pair 1. That's pair 2. That's pair 3. And that's pair 4. Okay, so 8 in total, 4 pairs of 2. Okay. So for this one, only 3 undergo crossing over, right? So crossing over occurs between only 3 of the pairs. So only 3 of these are actually going to do crossing over. Okay? So if this were to happen, this is an issue, and it's going to typically cause what we know as like a non-disjunction disorder, and you're going to have an unequal like split of chromosomes throughout, okay? So what's going to end up happening, okay. So for the first part where it's predict the number of chromosomes most likely, these are going to go into, right, you're going to do meiosis. So this is going to turn into 4. So let's just draw this. 1, 2, 3, 4, right? This is 1 gamete, 2 gamete, 3 gamete, and 4 gamete. You're going to have a non-disjunction disorder because 1 pair is not going to separate properly. For this, in the question, they basically are telling you that the crossovers and the double-stranded breaks are going to help with the distribution of chromosomes, okay? Okay, so if this were to happen, you're going to have a non-disjunction disorder, and basically, these ones down here are not going to separate from each other properly, okay? So as a result, what you're going to end up with is 2 of them are going to have 3 chromosomes, and then 2 of them will have 5 chromosomes. Okay. So this one will have 1, 2, 3, 1, 2, 3, 1, 2, 3, and then 1 from each of them. So basically, these ones over here took, again, you're taking half, it's meiosis. But these ones didn't separate, right? So this one is taking these 3, the other one gets the other 3. And then because those didn't separate properly, the other ones are going to have 5 in them. Okay, so 1, 2, 3, 4, 5. They're going to take both of these. So these don't get split equally because they didn't do crossing over. So both of these are actually going to be inherited together. Okay, so you're going to have, this one would be considered to be like N-1, so lacking a chromosome, and this one would be N+1, it gained an extra chromosome. Okay, so they should each have 4 in them because there's 4 sets of chromosomes. Meiosis should produce 4 gametes with 4 each. All right. However, we had a non-disjunction disorder between one of our sets, therefore 2 of them are going to have 3, and the other ones are going to have 5, okay? So you're never going to have 3 and 4 because the chromosomes have to go somewhere. So if these 2 gametes are missing a chromosome, that means these 2 gametes have to have the extra chromosome, okay? So hopefully this makes sense. This one's a little bit complicated because they don't really specify that it's a non-disjunction disorder. You kind of have to infer that because there wasn't a crossing over event that these didn't separate properly. So the actual answer for this one, let's write it down because it wants you to predict the number of chromosomes and then justify. So for D, I'll say 2 gametes have 3 chromosomes. and then 2 gametes have 5. Okay, and then to justify why, you're going to say that one pair is not going to separate properly, or it's going to experience non-disjunction. Okay, so this happened because cell chromosomes didn't separate properly because they missed crossing over, didn't separate properly. And I think that should be enough. Okay, so only in meiosis 1 is this an issue, hence why you're getting these numbers. So that is why in the case of these ones with the N+1, these ones took, again, both of those bottom chromosomes because they didn't separate. They technically had to travel as one unit. So those two traveled together. Imagine them stuck together and they have to travel together rather than being separated. That means that couple has to go to one gamete and then the other one gets none of those chromosomes. Okay? So that's why this one also has, obviously, 2. Okay, and then the last part is a little bit of an application. Explain how plant breeders can use the information in Table 1 to help develop new varieties of corn. So how would they use this to develop new varieties of corn? So let's look back at our table. Okay, so when we're trying to look at new varieties of corn, let's reread this. Plant breeders develop new varieties of corn are interested in determining whether in corn a correlation exists between the number of double-stranded chromatid breaks and the number of crossovers. Okay, so when you want to generate new types of corn, the one thing that you're going to want is genetic variation, right? You can't get new breeds of corn if all your corn is identical to each other. What does crossing over do? Crossing over introduces genetic variation. So if I am a plant breeder and I want to make a new variety of corn, I'm going to want crossing over because that's going to increases my chances of getting genetic diversity and getting, recombinant or, mixed genetic plants. So this is going to be good information. We know that the number of double-stranded breaks correlates positively to the number of crossovers. So if you're a plant breeder and you want more crossing over, you want more genetic recombination to make different plant strands, then you're going to want to find ones with a large number of double-strand breaks, okay? Because if I find the strain of corn that has a large number of double-stranded breaks, that means it's going to do crossing over a lot more, and I'm going to have more genetic variation. Okay? So for this, explain how plant breeders can use this information. So I would say that crossing over produces genetic variation. This is important to add because you want to know, well, why do I want to pick the ones that have more crossing over? You want to pick them because they're going to give you more genetic variation / new combinations, which is going to give you new variants. Okay, so crossing over produces genetic variation and new combinations, and therefore you're going to pick ones with most double-stranded breaks since they're correlated. Double-stranded breaks because positively correlated. Positively correlated. Okay. So I think they accepted 2 answers for these. I'm just going to read them. So crossing over, again, increases genetic diversity. They can breed strands with higher crossover numbers or double-stranded breaks, like I said. They can increase the number of double-stranded breaks, which leads to more crossovers. Okay, so that's actually interesting. That was the second option that you could have said, which I didn't even think of. So I would have just gone forward with the strains that had the most breaks because they're going to give you the most variation. Another option that they're saying that you could also say, you don't have to say both of them, just one. This is the first one that came to my mind, is that you could also induce double-stranded breaks. So you could have plant strains, sorry. Like let's say I want to grab this one, it has 710 breaks. I can break the DNA more because that's going to give me more crossovers is another way to do it. students probably aren't aware that you can, induce double-stranded breaks, but that is another kind of application to this. Okay, so either would have worked. And yeah, I think that's it for question 2, which was our other long one. I believe this one is also worth 9 marks. So in terms of breakdown, A is 1 point because it was just to describe. B was 4 points, 3 points for the graph, 1 point for the determining the difference. C was 1 point because you were just asking for the relationship, and D was 3 points. Okay, 1 for predict, 1 for justify, and then 1 for explaining the plant breeders. Okay, so that is it for number 2.